Celia and Flagella. So can you tell me what is the difference between Celia and Flagella? What is the function of Celia and Flagella?
The cilia are finger-like projections which will help in movement of substances.
Okay, then what
And
about Flagella?
flagella is a whip-like structure which is used for moving forward by any
Yeah,
bacteria.
yeah. So Flagella is found in both bacteria and eukaryotes, but Celia is found mainly in eukaryotic cells. Both of them are contractile filamentous structures which could be involved in movement. But the way in which they function, that is slightly different. And we have already studied flagella in case of prokaryotes in the second class. But this flagella in eukaryotes and the flagella that we discuss in prokaryotes, even though the function is almost same, the structure is completely different. They're structurally different. So cilia, they are usually smaller in size. And the way in which they work is different. They work like oars. So like imagine you are on a boat and you are rowing a boat. So similarly, that is how cilia will work. So this is called a power stroke. So it will move towards one side, then again it will move backwards. So like an oar, it will work. But flagella, you can see it will move like a snake. So this here is sperms of flagella, which help the sperm to move from one place to the other. The cilia is smaller in size, smaller in length, and flagella are comparatively long. But the number of cilia will be more compared to the number of flagella. Cilia will usually beat in a coordinated rhythmic manner, but flagella will beat independent. There is no such rhythmic manner. So the flagellar motion, we usually use the word undulatory motion. So undulatory motion is similar to the movement of a snake. Undulatory motion is shown in flagella, but in cilia, we use the term pendular stroke. So you've studied in physics about how a pendulum works, to and fro. So that is how a cilia will work. It will go back and come forth. So that is why we call it as pendular stroke. Cilia will help in locomotion, but it can also help in other processes like circulation, feeding, etc. But flagella is mainly involved in locomotion. So cilia is usually found attached to the cell surface. There will be so many cilia attached to the cell surface. For example, in your intestine, you might have some cilia, not intestine, sorry, in your this thing, respiratory tract, you have some cilia. So these will help in trapping all the dust and dirt particles. There might be cilia in your female reproductive system also, which will help in movement of egg or ovum. So you should be able to write the differences between cilia and flagella. Even though they are structurally similar, their functions are slightly different. So the internal structure is same, but external structure might be different. Cilia is shorter in size, flagella is longer. Cilia will work like oars, like pendular stroke. Flagella will show undulatory motion. Flagella are comparatively longer than cilia. And the flagella which is found in bacteria is completely different from the flagella which is found in eukaryotic cells. So let's see what is the internal structure of cilia and flagella. The internal structure is exactly the same. So if you take a cilia and flagella, cut it, take a cross section, internally it will look exactly the same. So internally, or if you take a cross section and put it under the electron microscope, you'll see that both of them are covered by a cell membrane or a plasma membrane. And they have a core region, the middle region, the main region is called, the core region is called axoneme. So the core is called axoneme, and this axoneme is made up of a number of microtubules. Microtubules are tubular structures which are found in cells. So a group of microtubules are arranged parallel to the main axis, and that is how it looks like. So you can see here, this is how it looks like under the microscope, a cross section of cilia or flagella. You can see small circles here. These are actually microtubules. So each axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules. So if you look at this picture, you can see there are two types of microtubules. There are some microtubules at the surface of the periphery. So that is called peripheral microtubules. And there are some microtubules in the center, two microtubules, one here and another one here. These are called central microtubules. So can you count the number of peripheral microtubules and tell me the number? How many peripheral microtubules are there? Now remember that one peripheral microtubule is this much. This much is one peripheral microtubule. So it is a doublet. Doublet means two rings. So count and tell me how many peripheral microtubules are there.
9.
Yeah, 9 peripheral microtubules. So it is called a peripheral microtubule. The word periphery means towards the surface. So it is almost towards the outer surface. That is why it is called peripheral. Why do you think it is called a doublet? Why is it called a doublet?
Because it is in a pair, I guess.
Yeah, because it is in a pair, there are two rings. That is why it is called a doublet. Okay, so there are two rings of microtubules, and that is why it is called doublet. You could call the first one A ring, and the second one is called B ring. What about central microtubules? Are these doublets? Can
No,
you say
sir.
that doublets?
No,
No,
sir.
no, right?
No,
They are
sir.
just
No, sir.
single circles, but they are connected to each other. So there are two central microtubules and 9 peripheral microtubules, which are doublets. Now, we'll go back to this. So there are 9 pairs of radially arranged microtubules and a pair of centrally located microtubules. So you can say that there are 9 on the surface and 2 in the middle. So that is why this arrangement is called 9 plus 2 arrangement of Cilia and Flagella. So what does this number 9 represent? 9 will represent peripheral microtubules, which are doublets, 9 doublets, and a pair of central microtubules, that is 2 microtubules here. Okay, so imagine these are microtubules arranged in the form of a circle. So imagine some long tubes, or imagine you have some around 9 to 10 straws with your plastic straw, and you're arranging them in the form of a circle with a gap in the center. So then that will look like these ring of microtubules. Okay, so each of the straw will be each microtubule, which will form a ring. So 2 such rings will form doublets, and there are total 9 pairs of doublets in this Cilia and Flagella. Also, you can see in the structure here, the central tubules, they are connected by bridge. So this is called the bridge, the green colored portion. This is a bridge. And also the central tubule is enclosed in a sheath. This red colored portion is called the sheath, a protective layer. It's called the central sheath. Now the central sheath is again connected to one of the tubules of each peripheral doublet by something called a radial spoke. You can see here, this is the radial spoke, another one, another one. So how many radial spokes do you think will be there in total?
9.
9.
9 will be there. Because there are 9 peripheral microtubules and there will be 9 radial spokes. So the function of radial spoke is to just connect the central sheath to the peripheral microtubules. Now, how are these peripheral microtubules connected? So you can see here, if you look carefully, you might see a green colored line here, this one, between these peripheral microtubules. So the peripheral microtubules are also interconnected by something called linker. So this is called linker. So linkers are found between peripheral doublets. So peripheral doublets are interconnected by linkers. So just below this diagram, you can see another picture here. So this is how it looks like under the microscope. You can clearly see there are 9 doublets on the periphery, and there are 2 microtubules in the center, which are the central microtubules. The radial spokes and other structures are not very clear here, but yeah.
So,
Yes.
so that interdoublet bridge is there. So
Interdoublet
that
bridges
and
is,
linker is same,
yeah,
no?
that and linker is same. That and linker are same things. So in your textbook, in the description, they have given linker, but in the diagram, they have given interdoublet bridge. So I'll show you. So in the textbook, they say, where is it? Yeah. So in the textbook, they say the peripheral doublets are interconnected by linkers. So linkers are the same thing, interdoublet bridge. The word interdoublet means between doublets. So the bridge between doublets is called interdoublet bridge. Okay. So understood the structure of cilia and flagella. It is a bit confusing, but it's actually very important. So cilia and flagella, they have a very similar internal structure. So what do you call the core of this? What do you call this entire part that you see? This entire ring, the 9+2 ring, this is called axoneme. Okay, it's called axoneme. So axoneme is the core of cilia and flagella, the main part. So axoneme has 9 peripheral doublets and a pair of central microtubules. And which one is protected by central sheath? Is it peripheral or central ring?
Central.
Central.
Yeah. The central one will be protected by a sheath. How many radial spokes are there? How many radial spokes
9.
are there?
9.
9 radial spokes. One second. Also, you can see here there are some proteins attached to this. These are motor proteins, and these motor proteins are called dynein proteins. So motor proteins are proteins which help in movement. The word motor means to move. So these are dynein proteins. You just have to remember the word dynein. We don't have to go into the details. Okay. So attached along each outer microtubule doublet is a protein, large motor protein, and this is called dynein. So dynein will basically help this microtubule assembly to produce bending movements of flagella and cilia. The cilia and flagella has to bend so that it can produce this either pendulum kind of movement or undulatory movement. So this dynein will help the cilia and flagella to basically bend.